So Dr. Brown has a PhD in genetics, genomics, and development from Cornell University. He holds a bachelor's in biology education from Pensacola Christian College and a master's in biology from the University of West Florida. So without further ado, Dr. Brown, welcome to the Creation Summit. Thank you so much for being here.
Thanks for having me. Hey, well, I appreciate this opportunity to share some of what we're doing on our own, but also to share on this really interesting topic in creation science at large. Let me share my screen. I'm going to actually be first. Let me just talk to the camera for a moment. I'm going to be jumping back and forth between screen share, and I have also some molecules that I'll be leaving the screen share. So there will be some moving back and forth as we go throughout this presentation. Just bear with me as we jump in. Let's get started. Today I'm going to be talking to you about a smoldering in the grave. Millions and millions of years ago, dinosaurs ruled the earth, and their reign of terror was brought to a catastrophic end when they had one very bad day about 65 million years ago when an asteroid impacted planet Earth, bringing the reign of dinosaurs to a catastrophic end after 180 million years. Or that's how the story goes. So this is all too common what you hear in every children's book, in every documentary, in every natural history museum. In fact, here, almost a direct quote from the UK Natural History Museum. They say how an asteroid ended the age of the dinosaurs. 66 million years ago, dinosaurs had the ultimate bad day with a devastating asteroid impact. A reign that had lasted 180 million years was abruptly ended. This is so common. It's everywhere. It's pervasive throughout the entire society that in order to believe otherwise, in order to believe that this whole age of dinosaurs did not occur millions of years ago, you would have to be just a little bit crazy. So permit me, if you would, for the next little bit to be a little bit crazy as we explore a smoldering in the grave. What decaying dinosaur bones tell us about the age of the Earth? So my name is Dr. Brown. You can call me Joel. You can call me Hey You. All of that works just fine. I got my PhD in molecular biology and genetics, also known as tiny stuff that you can't see without a very, very expensive microscope. I did that at Cornell University, had a fantastic time. I also spent some time studying Alzheimer's disease in fruit flies and spinal cord regeneration in zebrafish. So I've been blessed to have 8 children, and I recently moved to Arizona. So I lived in St. Louis for several years. I taught in the public school system for a few years in St. Louis area, and then I moved out here to Arizona with my family to become the director of the Creation Research Society. So this presentation is not about the Creation Research Society, but I do want to tell you a little bit about what we do. This is the largest network of Bible-believing creation scientists in the world. And it's just that. It's a network. So we have over 600 members who are degreed scientists. And they're at Christian universities, they're independent researchers. Some of them are in secular universities, kind of flying under the radar. Some of them are in institutes like the Institute for Creation Research. And the goal of this society is to connect all of these different individuals who have this shared interest in studying God's world, but studying it from a Christian perspective. And which brings me to one of my favorite verses, which I'm going to open up with Revelation 4:11. This is my motivation as a scientist, and the motivation of many of our scientists in this network would share it. It says, "Thou art worthy, O Lord, to receive glory and honor and power, for thou hast created all things, and for thy pleasure they are and were created." You can love God, you can love the Bible, and you can love the pursuit of science all at the same time. The difference is I want to give credit to whom it is due. So you just heard about all those incredible things that Salvador talked about, and it's sad, isn't it, to see these incredible things in nature these incredible structures, these engineering feats being attributed to just blind random chance over time. And as scientists, we can do better. Creation scientists, we're playing with a full deck. So that's our goal at CRS. We publish research. So we have our research journal. We present research. We have an annual research conference coming up in July. It's actually going to be in St. Louis. Check out creationresearch.org to get on that. We perform research. So this is a part that I'm heavily involved in here in the Phoenix area. We have a research lab. And so I'll be sharing for the first time, actually, some of the data that we've recently been generating in this lab. And we fund research. So if you have the next greatest idea in creation science, you can pitch that idea here to CRS, and we can potentially, if it's good enough, give you some funds to pursue that. So we want to generate an environment of science and scientific inquiry, but from a biblical perspective. So, all that to say, research is our middle name. And if you are not a member of the CRS and you are a degreed scientist, Bible believing scientist, you should be. I highly encourage you to join. I'm a new member. I joined just before finding out about the director position, and now here I am, and it's just been an honor to get more involved into the creation movement. So, with all that behind us, let's talk about a smoldering in the grave. So it's a bit surprising. I'm a molecular biologist, not a paleontologist, but this is a molecular biologist's foray into paleontology. One thing that is astonishing is how recently in the terms of human civilization, learned about dinosaurs, these incredible creatures called dinosaurs. So meet the first dinosaur ever named. This is Megalosaurus. And of course, they didn't know what it looked like, but British scientist William Buckland, he named it in 1824 based on this jaw fragment that they found. He noticed that this looked like it was from a lizard, hence the name Saurus, and it was big, hence the name Mega, and this became the first named dinosaur. The second named dinosaur was right on its heels, the famous Iguanodon. Same thing. British scientist Gideon Mantell. They found these bone fragments and teeth fragments, and they thought, well, this looks like it belongs to a reptilian organism like an iguana, but it was big, and so it was given the name Iguanodon. Now, did you notice those dates? I highlighted those dates on purpose. Those dates are a mere 200 years ago. In the grand scheme of time, that is not long. So here, human civilization is living all these millennia without an awareness of these giant creatures that existed at some point, and then they stumble across these dinosaur bones. The word dinosaur was coined just about 200 years ago in 1841 with Sir Richard Owens. So the discovery of dinosaurs is a recent thing. Now, of course, we have learned an incredible amount since then. I had the privilege recently to visit the SWAU Dinosaur Science Museum. This is in Keene, Texas. Here I am with Dr. Arthur Chadwick. I'd never been there before. It was a really impressive experience. These are Bible believers that study dinosaurs from a biblical perspective, and they have one of the world's largest collections of curated, geolocated dinosaur bones. Just an incredible amount of this. They go up to the Northwest U.S. every June, and they just dig for dinosaurs up there. They've been doing that for 25 years with the school there, and have been able to collect thousands of bones. So their collection has over 30,000 specimens in it. It's an amazing collection. Here I am holding a T-Rex tooth like that guy from the beginning. So all that to say, we have learned an awful lot about dinosaurs in the 200 years since they named the first one. Let me stop share here. Here is a ulna. So in your forearm, you have a radius and an ulna. The two bones that aren't supposed to be there, the two bone joint that Salvador was talking about. This is the ulna from a triceratops. So it's absolutely huge bone. We have a handful of dinosaur bones here at CRS. I'm gonna set this guy down. So all that to say, we have learned a lot in the years. I'm gonna get my screen share back up and running. Here it comes. Okay. And the question inevitably arises, so where does this all fit together? How where does it fit into a biblical timeline versus an evolutionary timeline? Well, it's pretty straightforward. Let's look at both of them briefly. Evolutionary interpretation of the fossil record is simply this progression of change over time. So they believe, of course, the simplest organisms were killed and laid down first, and progressively evolved into more complex organisms over time, including the dinosaurs in this Mesozoic era. And then those dinosaurs died off during extinction. In other words, in the evolutionary interpretation of the fossil record and of dinosaurs, they see a progression of time, an immense amount of time. So in the biblical creation interpretation of the fossil record, we also see a progression. We see an order. There is an order in these layers. There is a pattern that's recognizable throughout the world. And we do find dinosaurs in specific layers. We see a progression, but we see a progression of the flood. And I can't emphasize this enough. Isn't it amazing? Imagine the biblical record without the worldwide flood of Genesis 6 through 8. Imagine those chapters did not exist. And imagine that the Bible was the work of humans, right? So they just make up this record of the historic past, not knowing that these Near Eastern myths that they were writing would someday have to explain a finding 2,000 years later of these giant creatures in the fossil record. They had no awareness of this. And then they find these giant creatures, and lo and behold, you look in Genesis 6 through 8, and we have a mechanism that exists to explain how this entire fossil record is formed. So that's what we see. We see the progression of the flood in the fossil record. Now, this brings about two very different interpretations of when this happened. So when did the age of dinosaurs end, according to these two worldviews? So there's the age of dinosaurs, the Mesozoic. In the biblical timeline, these layers were flood layers. This is universally agreed upon by creation scientists that the Cretaceous, the Jurassic, Triassic, these were layers that were laid down by the flood. So, and that was about 4,400 years ago, give or take. So the age of dinosaurs came to an end 4,400 years ago. Now compare this to the evolutionary timeline. The evolutionary timeline says the age of dinosaurs came to an end with that catastrophic asteroid impact and volcanism around 65 million years ago. Right? Things that are different are not the same. These are very different claims about when the age of dinosaurs would have ended. And by the way, the age pre-flood would have been an age of dinosaurs. The amount of dinosaurs we find in the fossil record and the relatively fewer amount of mammals we see, there was a preponderance of dinosaurs pre-flood, which is an interesting question all to itself. But that certainly was an age of dinosaurs. So it then came as a surprise from a secular scientific perspective to find one day Mary Schweitzer and her research group was studying a T-Rex bone, and they found soft tissue. They actually sat on this data for a couple years because it was so controversial. Why would you find soft tissue in something that's 65 million years old? But here are some of the things that they found. This is a demineralized endosteal T-Rex. So they dissolve all the minerals away, and then you get the soft tissue that remains, including blood vessels and even things that looked like blood cells. This was understandably controversial at the time because these structures should not exist. They shouldn't have survived for 65 million years. So CRS, this was about 15 years ago, started the Dino project to look into this ourselves from a creation's biblical perspective. The iDino stands for Investigation of Dinosaur Intact Natural Osteo Tissue. That's a mouthful. I'll just call it soft tissue in dinosaurs. So one of the things that they did as part of this project was to go to Hell Creek Formation. That's where the T-Rex came from that Mary Schweitzer was working with in 2005 with their publication. So they went to the same formation in Montana in 2012. And here is a Triceratops bone. I showed you that Triceratops ulna. That is also from the same Hell Creek Formation. This was a Triceratops horn. 46 inches long, is encased in sandstone. And so some researchers here at CRS, this was before my time, they came and started analyzing to see if there was soft tissue in these bones. And they published this research in a secular peer-reviewed journal, Soft Sheets of Fibrillar Bone from a Fossil of the Supraorbital Horn of the Dinosaur Triceratops. So here are some of the images that they have from this Triceratops horn. And you look in these thin sections of the bone, these stunningly well-preserved osteocytes. Those are bone cells. That's what they look like in modern tissue, and this is tissue that even from biblical standards is thousands of years old, let alone the conventional timeline saying it's 65 million years old, and just these beautiful structures that looked fairly fresh. Here they are on scanning electron microscopy, and this again was published in another secular journal, Microscopy and Microanalytics. So these amazingly well-preserved bone cells. They looked at another bone. This was from a Thesalosaurus, the vertebral column, also from the Hell Creek Formation. So there's the vertebral column. And what you can do with these bones, you can take them and put them in this substance called EDTA. It's a chelator. It's a chemical that it goes in and it grabs the minerals from the bone, like the calcium, and it extracts the mineral from the bone. So you end up with just whatever's left that's non-mineral. So this is actual video from the Creation Research Society of the vertebra from this Thesalosaurus after it has been demineralized. And there is that soft, pliable, stretchy matrix of soft tissue. It's stunning that you can get this type of flexibility and preservation in these bones, even that are thousands of years old from a biblical perspective. Here is a video of what looks like a blood vessel coming from this Thesalosaurus vertebra. So these were findings right here in our lab in Arizona. Now, this has been replicated over and over again at this point, although it was highly controversial initially, and it still is in some circles. The claim, of course, is that, well, these tissues are not actual endogenous tissues of the dinosaur, but they're like biofilms that bacteria have formed later on. Well, now there have been over 100 papers that have been published describing the biological tissue and molecules in the dinosaur and other ancient fossils. And in these 100 plus papers, you see pliable tissue, like the video that I just showed you, distinct cells that are present in the tissue, like those bone cells that I showed you, collagen. And remember that word, that's going to be very important. Collagen fragments are frequently detected. Other proteins. So collagen is a protein. Other proteins such as actin, myosin, tubulin, they've also been detected. So the evidence just keeps building now that the secular community also, for the most part, has said, yeah, this is real stuff. Thanks in a large part to Mary Schweitzer. She's been very tenacious and diligent in demonstrating. She's not a creation scientist. She is just a researcher in the secular community. Although she is a Christian and would claim to be a Christian and seems like a very nice person. Thanks in a large part to her diligence, they've demonstrated beyond reasonable doubt that these are endogenous tissues, including here is the most recent one that I've seen. This is 2025. So this is fresh off the press. A paper, Evidence for Endogenous Collagen in Edmontosaurus Fossil Bone. This paper, there have been a number of ways that you can analyze what's in the bone to see if there's still protein and tissue present in the bones. And this paper does a neat one where they actually sequence the protein from the bone. So using mass spec, you can get the order of the amino acids that make up the protein, and they sequence the protein from the bone and identified it as collagen. So this is some of the first, not the first, but some of the first dinosaur proteins that have been sequenced. Now they're fragmented, so you're not getting high quality sequence, but you can identify it as collagen. All that to say, we have clearly this preserved tissue since these animals have died. I want to focus on this word collagen. I'm going to tell you a lot about collagen in the remaining talk. So let's unpack this a little bit. What is collagen? Now, if I were to ask you, what is a protein? What do you think of when you hear the word protein? Maybe you think of muscle and protein shakes and working out and amino acid supplements. And yeah, we often associate that with protein, meat, because our muscles have a lot of protein in them. But proteins are just these chains that build and assemble they comprise every structure in our body. So all of our cells have proteins in them. The muscle cells have a lot of protein, but so does your bone. So collagen is a protein. It is a chain of amino acids. See all those little red lines? Those are separating the amino acids. So we've got 1, 2, 3, 4, 5, 6 amino acids here in a chain. This is just a small fragment of collagen. Here is a full sequence of bovine collagen. This is from a cow. So that is, each of those letters represents an amino acid. So starting with M, and then you have a whole bunch. I want to zoom in on this. I want to challenge you. Do you notice? So each letter represents a separate amino acid, of which there are 20 that can be used in vertebrate proteins. Do you notice any pattern to this sequence? I highlighted 3 letters. That might help. That's a clue. This, do you notice how every third letter is a G? So this is the standard in collagen. Every third letter is G. That's a glycine. And then you have a couple other ones, and then another G. A couple other ones, another G. That's important for how collagen functions and its structure. So I'm going to do a little deep dive into collagen structure. I'm going to turn you, at least for the next 10 minutes, into little biochemists, okay? So bear with me. Buckle your seatbelts. Let's start with this guy right here. This little black ball is a carbon. It's a carbon atom. Okay. We are called carbon-based life forms because we are carbon forms the backbone of all these major biological molecules. Okay, let's start adding some things. We're going to add a hydrogen and another hydrogen. Now we're going to add, so now I've got a carbon with 2 hydrogens. Now I'm going to take this guy. See how that's kind of a blue? So this is nitrogen. It's an amino group. I'm going to add that. And then let's add another group onto that carbon. I'm going to add this. This is a carboxyl group. It's a carbon with 2 oxygen. You don't need to know all that, but and voila, I have created before your very eyes an amino acid. This is one amino acid. This is actually glycine. So this is the glycine, which is in every third amino acid in collagen. And that's what it looks like. Okay, so now there's not just one amino acid, there's actually a long chain of amino acids. How does that work? All right, I'm going to take another amino acid. This one is alanine, similar in structure. There's one small difference, which I won't bore you with. Okay, now let's try to stick these together. So in order to do this, I'm actually going to remove one of these oxygens. And here comes my alanine. And now we have an amino acid chain. This is 2 amino acids long. All right, and the reason I'm showing you this is going to be very important later on. Do you see this purple bond there? I colored that one purple on purpose. That one's very important. It's called an amide bond or a peptide bond. Keep an eye on that one. Okay, let's just do one more. So we've got our glycine, we've got our alanine. Let's remove another one of these. This is a hydroxyl group. And we are going to plug in a third amino acid, like so. So now there's one purple bond. Here's the second purple bond. Those are the peptide bonds that separate these 3 amino acids. And there you have a beginning of a collagen chain. So to go much further would get very complicated, and I don't have enough hands to hold all those. Let me try showing you this in some software. All right, perfect. So here, this is just a protein modeling software where you can visualize these things. This first one that I'm showing you, this is glycine. Now we have a long chain of them. Here's the full chain of collagen. But collagen is not just one chain, and this is important for the structure of collagen. Collagen is actually a group of chains. So here's where we started. This is what I built for you. Was this part here in the middle, and the full length is the full protein. This is one collagen protein, but it's actually not just one, it's 3. So now you see how there's 3 different colors. There's the green collagen protein, there's the blue collagen, and there's the pink or magenta one. Well, that is the structure of collagen. It shows up in this helical twist of 3 collagen proteins. And here's what it would look like. It's 3-dimensional shape. It's these long strands. So now you have been christened as biochemists. This is the structure of collagen. I'm going to stop that share. Let's go back. Okay, there we go. So that's the structure of collagen. Here's why that's important. That is actually an ingeniously designed structure to provide stability. Collagen is a structural protein. It's in your skin. And when the collagen starts to degrade, you start to get wrinkles. That's just a fact of life, right? Collagen is a structural protein. And so what does collagen have to do with bone structure? A lot. So bone is a mixture of minerals. That's the inorganic part, the calcium, phosphorus, and protein, like collagen. Collagen is actually the main protein in bone. So here's a diagram of bone. This is like a cross section through the bone. Bone is composed of 60% of this bioapatite. That's the mineral component, 10% water, and 30% organics. What's organics talking about? Well, those are the proteins. Out of those organics, it's 20%, 27% collagen. So 90% of the proteins inside of bone is collagen. Collagen is the most abundant protein in bone, and it is the most abundant protein in your body. So it's a very important structural protein that impacts all of our biology. Now to the question at hand. How long can collagen, which has been found in dinosaur bones, how long can collagen lie smoldering in the grave? So I'm going to take you on a little journey on how our lab has been investigating this very question through an experiment. So this is examining collagen decay in different organisms. We, of course, we're using modern organisms, so we can start with a fresh bone and then see how long the collagen survives in a fresh bone. You know, when the dinosaurs died, they would have been fresh bones and would have started to decay. So we're doing this in mammalian bone, a cow. We're doing this in avian bone, an ostrich. And we're doing this in reptilian bone, an alligator. And I know what you're thinking. Where do you get alligator bones from? From alligator legs, of course. So here we are in our lab. We actually just got a shipment of alligator legs a couple of weeks ago. We just asked this alligator hunter to send us the biggest legs from the biggest alligators that he had. And he sent us some big ones. I was surprised. And so here we are dissecting those legs, pulling out the bones. We are in the middle of doing this with alligator. The results I'm going to share with you today are from cow and ostrich, and I haven't shared them. This is the first time they've been shared publicly, so I'm excited to let you guys in on what we're doing. So remember, collagen is a protein, a chain of amino acids. So the question, we have to find a method. We have to have a method in order to see how this protein dissolves or breaks down. And when we say break down, what we're talking about is those peptide bonds, those purple bonds that I showed you, those break apart. And it's water that gets in there and breaks those bonds apart. That's what we mean by degradation. So I circled the 5 peptide bonds in this protein, and that's what we're studying. So how do you tell when a peptide bond has dissolved? There are several ways you can do that. The way that we use, the method we use, is Fourier transformed infrared, FTIR, spectroscopy. What in the world is that? Here's what it is. It's a very, very expensive flashlight. Literally. That's what it's an expensive flashlight, but it's a cool flashlight because it shines in the infrared spectrum. So it's not visible light. It's longer wavelengths of light. And you can shine it at a sample. And then depending on how much light comes back to you, you can begin to identify that sample. So there we are in this example over here. Here's our sample. We shine in this light source. It beams through the sample, and then you have a detector. It's pretty complex what's happening in the machine to adjust the wavelength. There are moving mirrors and stationary mirrors and all that good stuff. All you need to know is we're shining a flashlight at a sample, and we're seeing how it reacts. So here's an example. This is 2 amino acids stuck together with this special bond, the amide bond or the peptide bond. Same thing. We are now shining this beam of light, infrared light, at this protein. And those atoms in there, the oxygens, the carbons, the nitrogens, the hydrogens, those bonds between them are going to absorb some of that light, and some of the light gets transmitted. It gets passed through the sample. And you can detect the presence of that bond, the amide bond, based on how much light is passed through. So that's what we're doing. Here's an example from the literature. A very recent example. We're not the only ones trying to pioneer this technique. So here, the green line represents well-preserved bone, fresh bone. The red line represents decayed bone. So it's older bone that's gone through decay. And I want you to pay very close attention to this mountain peak right here. You notice how in the green line, it's very tall. In the red line, it's very small. So these mountain peaks come from the machine. This is the data that comes out of the FTIR spectrometer. And this is the mountain that we are looking at. It's a very specific mountain. This was pioneered also by Brian Thomas at Institute for Creation Research and others. Kevin Anderson was the former director, so they were tinkering with this. Like, how can we use this to measure collagen decay? So again, look at this mountain. Here it is at day 0 in fresh bone. It's a pretty tall mountain. At day 14, it's a pretty small mountain. That's what we're looking at. And you can learn a lot just by looking at that mountain. Here's an example from our lab. This is data we just were working with last semester. I'm going to zoom in on this mountain peak. This is what came from the FTIR. And here is the mountain peak that we're interested in. You notice how at the blue line at time 0, it's a tall mountain. Times 7, it's a smaller mountain, and it continues to decrease. So we can measure the height, and that tells us how much collagen is present. And you notice this is important. This peak here, which is the mineral component, it doesn't change. So the bone is just being bone, but the protein is degrading. All right. So that's the tool we can use to measure collagen decay. Let me show you how this works. This is what we call our materials and methods, or our experimental design. So we're taking a bone which has collagen in it. All bones do, and a lot of it. We crush up those bones into tiny fragments. We then are accelerating the decay because these are extremely stable proteins. From a design perspective, they're designed to be extremely stable. We're not going to wait around hundreds of years for them to decay. So we're going to speed it up with temperature. We grind those bones after they've been treated with temperature. We grind them to a fine powder. We then put that powder onto this FTIR and we scan it. We get our mountain peaks. We measure those heights, and we do this at multiple temperatures in order to generate these decay curves. And we can put all that together to generate these things called Arrhenius plots. I'll show you the Arrhenius plot, and I'll explain it. I'm going to jump right to the end, and then I'm going to back up and kind of explain what this is showing you. So for now, don't worry about everything on this slide. For now, I just want you to I want you to notice this was the end of the experiment. This was the final result from about 6 months of working with these bones. I really want you to appreciate kind of the back end of scientific research. So I'm going to show you just a short video of how we did this so that you can appreciate exactly how much goes into a single image like this, a single graph like this. Sit back, relax. I'll narrate through it a little bit. Here we go. Those were the alligator bones. We crush up those bones into smaller fragments. We continue to clean them and crush them ever smaller. We want to get nice uniformed size in order to have kind of repeatability and reliability in our data. So we continue grinding it till they're this nice size, about 0.5 millimeter to 1 millimeter in size. Then comes the fun part. We start cooking our bone. What you're seeing here is we're taking 1 gram of our bone sample. We're mixing it with some water. So this occurs in aqueous environment, mimicking the groundwater. That's during fossilization. That's how fossilization occurs, is exchange of minerals in the groundwater. Now, again, because decay takes so long, we accelerate this decay by increasing the temperature. So that's what you see here. We do this at multiple temperatures, and we're just showing you one example. Now, we're going to be testing how much has decayed at different time points. So we're recording how much time has passed between each pull. Now, here comes the first pull. By the way, these are undergraduates from Arizona Christian University. They interned in our lab. So once they've cooked for the specified amount of time, and of course we're doing multiple time points, we do 3 replicates at each time point. We now have to prep the bone for measuring. There's the bone. It's still hard bone at this point. It's just small pieces. Now, of course, we are not doing this with just 5 samples. We're showing you 5, but there are many, many, many samples that we have to do this with to repeat the experiments for replicates and all that good stuff. So now we have to grind that decayed bone into particle sizes that are appropriate for the FTIR machine. The particle size that it needs is between 20 millionths of a meter and 63 millionths of a meter. So we use these sieves to screen it down to just the right particle size. So there's the bone powder. I know what you're thinking. It looks like drugs. It's bone powder. And now we're ready for the next stage, which is the measurement. This is Ryan. He's a part-time tech in the lab. He's really the professional at the FTIR. He's been doing it for several years now. So the FTIR is a surprisingly simple process. There's the powder. And then the machine does all the hard work. It shines a beam through there, measures those mountain peaks, and that's what the output looks like. We measure each sample 3 times. Those would be called technical replicates, and we do this over and over again for every temperature, for every time point, for every organism, and we get a lot of data. So this is combined data from ostrich and cow. The final step then is to go to the computer. So we don't want to analyze this data ourselves. We do it using stats programs. So this is a program called R. And we use the code to then analyze, look for patterns in the data, and generate these plots. All right. So I hope you see why I showed you that. This is whether you're a secular scientist, a Bible believing Christian, whatever, you can do science. This is called empirical science. It doesn't matter what your worldview is. The bones are going to decay the same, regardless of whether you have a biblical worldview or a secular worldview. This is just good old fashioned empirical science, and it takes a lot of time. It takes a lot of money. It takes a lot of patience to do it. So now let me show you the results of those months of labor. All right, so keep the question in mind. How quickly does collagen decay? So here is ostrich at 85 degrees Celsius. I'm showing you the collagen concentration on this y-axis. I'm showing you the time in days on the x-axis. And so it starts high and it decreases to low, and it follows this nice linear pattern. That's one temperature. Now we want to do this at 5 different temperatures. So this is 70 degrees. It starts high and it goes low, but it doesn't go as quickly, right? Because it's a lower temperature, so it decays more slowly. The breaking of those bonds is temperature dependent. It's depends on thermal energy. And so as you increase the temperature, you notice how the slope gets steeper. And that's because it's decaying faster, as you would expect. This is thermal energy. Okay, so now let's compare this to the cow. We've got the ostrich on the bottom. We've got the cow on the top, same setup as before. And they both follow that similar pattern, slower decay at lower temperatures, faster decay at higher temperatures. We're doing this at high temperatures because then we can calculate what it would be at a lower temperature without waiting hundreds of years, because this takes hundreds of years at lower temperatures. So here is how we do that calculation. This graph is different from those other graphs I was showing you. This is now showing you temperature on the x-axis and the decay rate on the y-axis. So you can see at higher temperatures, like at 90 degrees, we have higher decay rates. At lower temperatures, you have a lower decay rate. Here's the decay rate at 85. Here's the decay rate at 75. And you see how there's 3 spots for each of these? That's because we're doing this in replicates. So we have triplicates for all this data. And once we build this curve, this is the curve that we were working for all those months, because now we can see what would the decay rate be at lower temperatures based on this curve. And here it is. Here, this is the answer. Half-life at 25 degrees Celsius of collagen in a neutral aqueous environment is 1,086 years, give or take. That was the result from our experiments. A half-life, remember, is how long it takes for half of that collagen to disappear. So after 1,000 years, you're going to have 50% of the collagen remaining. After 1,000 more years, you're going to have 25% remaining. After 1,000 more years, 12.5, so on and so forth. So that is a very relevant and important result. By the way, here it is, the cow, which I showed you, and here's the ostrich. We were expecting them to be basically the same. It's collagen, right? It should be about the same. They are different, actually. The ostrich decays significantly faster, has a half-life of 296 years at 25 degrees Celsius in a neutral aqueous environment. You might be wondering why we are doing ostrich. Well, because as you know, evolutionists, they would call dinosaurs non-avian. Or birds are avian dinosaurs. The dinosaurs are non-avian dinosaurs. So they call birds dinosaurs. And so in many of their work, they compare their collagen stuff to ostriches. So we want to meet them on their turf. So that's why we use ostrich. But we're also doing this in alligator, and I'll report back to you on that once we got those results. So there you have it. Now here's what was very exciting to us as a lab. You do these results, you do these experiments, it takes a long time, and you hope that they are relevant data. So here's another paper that was published, Collagen Survival and Its Use for Species Identification. They studied collagen decay using a different method, and here were their findings. Extrapolation from high temperature, that's exactly what we did. Experimental decomposition rates suggest that at a constant 10 degrees Celsius, it will take between 0.2 and 0.7 million years at 10 degrees Celsius for levels of collagen to fall to 1%. Do you see how precise that statement is? And that's so that other scientists like us can compare our results to it. So let's do that. Let's compare our results to theirs. Here's our data. Our data says that the half-life at 10 degrees Celsius, because we can calculate it at any temperature. I was showing you at 25 degrees Celsius, which is room temperature. 10 degrees Celsius is pretty chilly. The half-life is 35,000 years, half-life of collagen. 7 half-lives, which is how long it would take to decrease down to below 1%, 7 half-lives. That's 250,000 years. Now think about what that is saying. Our study, their study, showing the same thing. This is just empirical science. This is the way science is supposed to work. It's repeatable, it's observable. But what do you notice? The longest we can reasonably expect collagen to last under ideal conditions is approximately 250,000 years. And I say ideal conditions. Here's what I mean by that. We don't have these huge temperature fluctuations, which would be bad for collagen degradation. We don't have bacteria in here. Bacteria decay collagen even faster. So this is an artificial setting. This is kind of an idealized setting for collagen decay. And there's our ceiling, 250,000 years. So how long can collagen lie a smoldering in the grave? I'm so glad you asked. Not 65 million years by conservative empirical estimates. Now, as a scientist here, did we prove that collagen could not survive 65 million years? No, that's not the way science works. But what we did demonstrate is that a reasonable estimate for how long collagen lasts is that it's going to all be gone well under 1 million years. All right, so what do you do with this? Well, this is a problem, as you can see, for a secular evolutionary worldview, and they recognize it. Here's Mary Schweitzer. And she says, the persistence of still soft vessels, matrix, and cells and fossil remains millions of years old is unexpected. And if original, refutes models of, what do you think goes in that blank? So if this is original material, it refutes models of, maybe she's about to say, of the age of the earth or deep time. Or no, it, in their mind, refutes models of degradation. So I guess things just don't degrade that quickly, even though we have empirical data showing that this is how it does degrade. The identification of still soft tissues and cellular structures in a suite of Mesozoic fossils and claims of endogenous proteins preserved within these materials is controversial because it challenges both conventional wisdom and theoretical kinetics. That's our data, which preclude the persistence of proteins over geological timescales. So they recognize this. This is 2019, so it's still an issue, and they have proposed models that try to address this. Here's the cross-linking hypothesis. This is the main one. They say that iron from within the body of that dead animal in the hemoglobin, myoglobin, it helps to preserve it. Here's what they're talking about. That iron causes these hydroxyl radicals to form. That's what those O's and those H's are. And those hydroxyl radicals, they bind to the amino acids and they cause this cross-linking. And the cross-linking is thought to resist the degradation. So it's a more stable protein. It can last longer. Well, this is good, right? This is a hypothesis that we can test. And so that's what we're working on next in our lab we've got a nice system for measuring decay. Now let's test these hypotheses from an empirical perspective and see if it really can extend it to 65 million years. Stay tuned. We'll let you know what we find. We're starting these experiments now. However, it's not very optimistic because here is, for a secular evolutionary perspective, here's another group that tested this already. They took bone and they have a cool setup here. They have little bone samples in all of these tubes, and then they run different solutions through there. So one of the solutions that they put through these tubes was iron, and they were, presumably expecting it to extend the degradation. Here's what they found. The iron actually accelerated decay in their tests. So iron isn't going to be the savior for this problem. There are many, many saviors that have been proposed by how to extend this preservation of the collagen in spite of the empirical evidence that we see that says otherwise. And I don't want us to get lost in the whack-a-mole game of trying to knock out every latest and greatest idea, because you can knock out the iron hypothesis, there's going to be another one. There's always going to be another one because this is historical science. This is not empirical science that evolutionary scientists are doing. That's just the nature of it. And you can see it broad and clear right here. This is from Schweitzer's 2007 paper on this topic. If these components are demonstrated chemically to be original, a mechanism must exist to allow their persistence across geological time. You can call that a lot of things. You can call that bias. You can call what it is just a commitment to a worldview. They see no other option. The time exists, so there must be a mechanism to preserve it. Maybe the time actually doesn't exist. So let's be very clear here at the end. When did the age of dinosaurs end? Biblical timeline says 4,400 years ago. Evolutionary timeline says 65 million years ago. Things that are different are not the same. Dinosaur biomaterial is not consistent with the conventional assigned ages of the fossils. That's what our data shows. Dinosaur biomaterial is consistent with a younger than 10,000 year ages of the biblical model of creation. So isn't it exciting sometimes when, we aren't trying to prove the Bible, right? If God wanted to prove the Bible, he would do that himself in an empirical way beyond any measure of doubt. We're not trying to prove the Bible, but it is fun as you're a scientist pursuing the study of God's creation, and you see these things that support just a straightforward, simple reading of God's word. So I want to finish again. Our objective is to give God the glory. Thou art worthy, O Lord, to receive glory and honor and power, for thou hast created all things, and for thy pleasure they are and were created. Let me finish by giving credit to whom credit is due. I want to thank our team here, and it's not just our team. We're standing on the shoulders of others. For instance, Brian Thomas at the Institute for Creation Research, Steve Taylor, other individuals who have pioneered some of the early steps in this work. And I, of course, want to thank the work here at CRS and in our society at large is supported by generous donations of God's people. And so without those donations, this work wouldn't be possible, and I thank you for that. with that, I'm going to stop there. Thank you for your attention and turn it back over.
All right, Dr. Brown, that was awesome. Now we do have quite a bit of questions. So it looks like you spurred up some curiosity. I'm going to mix one of them kind of together.
Go.
So it's questions about glycine from Solidify Your Face. How did glycine get into Comet Wild 2? And can glycine form naturally in space or on Earth? Additionally, 14 of the 20 amino acids used for life on Earth have been found on asteroid Bennu. Would CRS consider the hydroplate theory for an explanation?
There's a lot in that question.
Yeah,
So let me just speak to what our data is showing. Um, Glycine, and we take all of these amino acids to be a direct creative act of God, whether they're on other asteroids or not, why they would be. They're just molecules, right? So there's really nothing special about them. They can be anywhere in space. That's fine. That doesn't mean there's life out there. They're just dead, lifeless chemicals. What's amazing here on Earth and in God's creation is how those molecules, how those amino acids are arranged. Glycine in collagen is a perfect example of it. So glycine is the smallest amino acid of the 20. I showed you that the structure, it only has this one little hydrogen on the carbon, which makes it the smallest one. And so it has to be there in collagen in order for that collagen to twist into that beautiful three-dimensional structure. And so that's why glycine is so important in it. As far as the hydroplate implications of that, that's outside the scope of this study.
sure. Wait, would you know the half-life of glycine?
Oh,
Probably not.
so glycine itself, I'm talking about the half-life of the peptide bonds, which is the bond between two glycines. And even for two glycines, the half-life is somewhere around 500 years.
Okay. And then we have from TH Hale. What is the relationship to humidity? I believe he was talking about during the testing process. You did heat, you did other things, but would moisture in the ground maybe affect the half-life of the collagen as well?
Good question. Yeah, so in our upcoming, now we've kind of built a baseline and a method that can robustly measure this. So those are some of the things we want to look at. Um, Water definitely has an impact. So how much groundwater is present, and or in your case, what this person is asking about, the humidity of the environment. That water is what's getting in there and breaking these bonds apart. So there has to be water present. If you had a completely dehydrated sample, yes, you would extend the decay. So that's something we want to try is how if we dehydrate it. Now that's unnatural conditions because fossilization is occurring because there's groundwater there. So that's an unrealistic situation, but we still it's a valid question and something we want to look into.
Are you still working with Mark Armitage and his electron microscope?
I I wish in that he is a very talented microscopist. I've not met him, and I do hope to meet him at some point. As far as a creation microscopist goes, he's as good as it gets. Um, and so he has a soft tissue, like Dinosaur Soft Tissue Institute, something like that. And he's still doing this type of work and getting really neat images out of it. So as the opportunity arises, I would welcome the opportunity to work with him on that.
And then last one, are there any, this solidify your faith, are there any creation teaching churches in the Phoenix area?
Well, I'm at one. So there are several. I'm attend a Baptist church in the Phoenix area. It's a growing area, as you probably know, and so there are actually a good body of just faithful believers in the Phoenix area. So there are many churches that would adhere to a straightforward belief in God's word.